is maintained extremely precisely. It's just scrupulously regulated. And that's kind of good because I should say it's rather fortunate that you have a calcium repository in your body. What's your calcium repository? Yeah, the bones, the teeth, and so on. But of course, physiologically, homeostatically, your body places primary importance upon the ECF calcium, and that bone is going to be secondary. So that's really what we're talking about here, the mechanisms by which the body regulates this ECF calcium. Arguably, it's the most abundant mineral. Not sure that that's true under all circumstances. In the skeleton, it's present, at least in humans, it primarily is calcium phosphate. There's some other calcium salts that are present in bone and dentin and other tissues as well. And we've already pointed out that it's that ECF component that is most critical. Calcium is going to be frequently bound. It can be bound to proteins. It can be bound up in salts. So as was our warning with the hormones, it's the free calcium, it's the free ionic calcium that is most important. And in adults, a normal adult, ECF calcium is on the order of 9 or 10 milligrams per deciliter, a little lower than that for children. I mentioned children because in your practice where you're going to see perturbations of calcium, and we've talked about hypocalcemic tetany, we've talked about hypercalcemia, that tends to show up more commonly in children than in adults. So I've already mentioned some of these critical functions. Just about every aspect of physiology involves calcium to one degree or another, and either directly or indirectly. So make sure you review that list if necessary. The ability of calcium to be shuttled between the bone and the ECF is critical. We're going to be using several terms to describe that process. But remember, there are going to be hormones that basically regulate the extent to which bone is sacrificed on the behalf of ECF calcium and vice versa. So we're going to be looking at those hormones in more detail. I think you recall the terms we've used in the past. Remember we said in the past that bone is remodeled, bone is constantly remodeled through the activity of osteoblasts and osteoclasts. Osteoclasts resorb bones, and notice the spelling there. It's not reabsorb, it's resorb. Resorb means to break down, degrade, and utilize in some other fashion. The osteoblasts, on the other hand, are responsible for deposition of the bone matrix. So you have this antinomial balance between the activity of the osteoclast and the osteoblasts. Now, I've indicated the way that calcium is present in the circulation. It can be bound either to proteins or to salts, but it's that free form that's most abundant. I don't see a percentage there, but in general, about half the circulating calcium is in the free ionic form. So the rest is either going to be bound up with protein or with salts. And we don't need to worry too much about the dietary calcium. Generally, most in the American diet, it's more than abundant. But be careful of the terms again, because we're going to be using similar terms in very different ways when we talk about, and we will be when we talk about calcium uptake or calcium absorption. We're talking about GI tract absorption. We're going to find, for example, that vitamin D plays an important role in regulating the uptake of calcium from the GI tract. We've talked about the components of a long bone. It's important to keep in mind that the bones depend upon adequate calcium salt, adequate calcium availability, and that certainly in the absence of adequate calcium, or in the event that there's a derangement of these hormones, these endocrine regulatory mechanisms that allocate calcium between the ECF and the bone, that one of the consequences could be osteoporosis, a general loss or diminution in the proportion of calcium that is present, the calcium salt that is present in the bones, especially the long bones. We've already talked about endochondral ossification. I just want to let you know that calcium and all of these calcium regulatory hormones that we will be talking about are essential for normal growth, the normal elongation of the metaphysis. Remember, this is the diaphysis. This is the primary ossification center, and as the bone elongates, you have a cartilaginous zone in here that migrates toward the epiphysis of the bone. And in order for that ossification to occur, there must be adequate not only calcium, but the levels of vitamin D and PDH and so on must be appropriate. All right, so we've already talked about these three main characters, these three main actors in the cells that are present in bone: the osteoblast, osteoclast, and osteocytes. It is generally considered the case that the osteocytes, the mature bone cell, arises from osteoblasts. The osteocytes become entrapped in the bone matrix. It's as if the osteoblast deposits the matrix around it. At that point, it can no longer migrate and becomes an osteocyte. In the course of ongoing bone remodeling, osteocytes are frequently sacrificed. They're frequently lost and then regenerated through that whole process of osteoblastic activity and osteoclastic activity. So it's, again, this sort of dynamic system. And even as you're sitting there right now, your bone is being actively remodeled. If that doesn't happen, then the bone becomes more brittle, loses its flexibility, loses its vitality, and there can be adverse pathological consequences. In the figure here, what you'll notice is we have something along the lines of a progenitor cell. The term we've used to describe such cells in the past is multipotent or oligopotent stem cells. Remember the use of that terminology earlier, multipotent or oligopotent? It means they're stem cells, but they're only marginally so. They can only differentiate with few types of cell. And by differentiation, we mean that these stem cells can become, for example, chondrocytes. Or, as you can see, that same stem cell gives rise to a pro or pre-osteoblast and even a fat cell. And we're not too concerned about the adipocytes now. And this is going to be important to our story later, but notice that from that stem cell, you have sort of a precursor osteoblast. And these precursor osteoblasts give rise to sort of precursor osteoclasts. And so I guess the point that I would make here is that in this lineage, this cellular lineage, we're going to find there must be progressive differentiation from the progenitor cells into the blasts and the clasts. And you can see that even the osteocyte arises from that lineage. So it's all the same sort of lineage. But more importantly, as we talk about this in just a moment, when we talk about endocrine regulation, it's essential to understand, I guess the way I would put this, is it's the osteoblast that determines osteoclast activity. Let me say that again. It's the osteoblast that determines osteoclast activity for reasons that will become clear in just a moment. I think we've looked at illustrations like this before in the micrograph here. You can see this large multinucleate osteoclast. I'm not sure it's evident up here, but what tends to happen, these osteoclasts start out as small cells and then they start fusing together. More of these precursor cells start fusing together until eventually you get this large, you can see it up there, it looks like one of those scrubbing bubbles that you saw on the advertisement years ago. With the ruffled border, there's the ruffled border, and this thing releases enzymes, degradative enzymes that can erode the bone. But really that osteoclast that you're looking at in the upper right of that image is the fusion of multiple smaller cells. And so that's really what we're looking at here. We're looking at a cell down here that's really represents the fusion of multiple precursor cells. And then you don't see them so evident in this illustration, but oftentimes ranging in more than one direction from the osteoclast are multiple osteoblasts. And then this system, this phalanx, if you will, of osteoclast and osteoblast are just sort of eating away bone. And as the osteoclast eats away the bone, right behind it come the osteoblasts that redeposit fresh bone matrix. I indicated a moment ago that it's the osteoblast that primarily determines osteoclast activity, and that's because there is a far greater number of receptors of all types on the osteoblast than on the osteoclast. And this is important. It seems trivial, but this is important for you because a lot of the medications, a lot of the therapies that you'll be using to address pathologies, osteopenia, osteoporosis, and so on, Paget's disease, require an understanding of this fact that you're really, even if your target is the osteoclast, in most cases you have to work through the osteoblast. So yeah, I indicated these osteoclasts are large multinucleate cells. You can see the ruffled border down here from which hydrolytic enzymes are released. These enzymes are often released in a soup of a low pH type of corrosive juice. This gap down here that's created by the osteoclast is a lacuna, sometimes called Hausch's lacuna. And there are all kinds of, as I indicated, all kinds of pathologies associated with some defect, some derangement of the concerted activity of the osteoclast and the osteoblast. This whole system of osteoclasts and osteoblasts goes by a variety of names, at least in the literature. I've seen quite a few terms. One of the terms you sometimes see is the basic multicellular unit, and that term simply describes what we're looking at up here, sort of a phalanx of osteoclast with peripheral osteoblast moving through, tearing through the muscle, eating away the old and redepositing the new. Yeah.
So when there's a low concentration of calcium ion in the blood, is it always, are the osteoclasts always removing bone from the spongy bone, or is it, do they sometimes stay from more?
Yeah, it's essential to understand that all of your bone, including the compact bone, including the paracentric bone, is being remodeled actively. And if you think of those aversion systems or osteons, those are constant. I mean, one of the reasons, if you look at the illustrations of them, you've got concentric rings on top of other concentric rings. It's because all of that's constantly being remodeled throughout your lifetime. Now, as I say, there are several hormones, and the literature is a little bit equivocal on the relative importance of these days. But there are several hormones listed here that play an important role in calcium ion regulation in the ECF. Most important of which is PTH, parathyroid hormone. I bet you can guess where it originates. Sometimes called parathormone, but we'll just call it parathyroid hormone, or PTH. Again, it is a peptide. You want to pay attention to the structure because the structure informs you about the mechanism of action, transport, and so on. So we'll spend some time talking about PTH. Another is vitamin D. I think we pointed out earlier that vitamin D and PTH are permissive with respect to each other. And there's a lot of debate about calcitonin. It depends on whether you want to take a conservative perspective or maybe a revisionist perspective. But I think most of the recent literature suggests that calcitonin has other things to do. It's probably more involved in other physiological processes, and the traditional attempt to give it a great deal of responsibility with respect to calcium regulation is probably overwrought. Probably calcitonin is less important for calcium regulation than we previously believed. If we have time, we'll come back and talk about that a little bit. These hormones, of course, are going to be targeting the bone, the GI tract, and the kidneys. So make sure you appreciate those three general sites. The PTH in particular targets the bone through modulating or regulating the rate of remodeling of the bone. The vitamin D in particular regulates to a large extent the intestinal uptake of calcium, and both tend to play a role in the extent to which calcium is either emptied into the urine or retained by the kidneys. So PDH, we talked about the location of these typically 4 parathyroid glands on the posterior side of the thyroid. Important for you to understand anatomically, because if there is thyroid surgery, sometimes these get involved, and if these get involved, that's going to disrupt normal calcium regulation. If thyroid disease of itself can sometimes involve these, a tumor, cancer, whatever, can sometimes involve these sort of anatomically substituent structures. Now, the general rule that you want to keep in mind is that parathyroid hormone is released from parathyroid glands in response to circulating calcium ion concentration. So that means there must be something in those parathyroid cells that has the ability to monitor circulating calcium. And we'll talk a little bit about that pathway in just a moment. There's some other things that stimulate PDH release, for example, magnesium, the sympathetic nervous system, and glucocorticoids, but we're primarily going to focus upon PDH levels changing with calcium. So what should happen is that as ECF, as blood calcium levels go up, you would expect PDH levels to go up, right? And so depending upon which end of this you approach, from the hormone to the element or vice versa, we can say that there's normally an inverse relationship. In other words, if PDH levels are high, what's the effect? I guess I don't think this is clear, so let's make sure we understand the effect. The effect of PDH is to take calcium from the bone and empty it or place it into the circulation. So, in fact, both of these hormones, the only exception being calcitonin, is they're going to act to increase blood calcium. So there will be several points in the course in which maybe it's simpler to just think about 2 compartments. With respect to calcium, we have the bone and we have the blood. PDH is taking calcium from the bone and increasing the concentration in the blood or in ECF. So if that's true, if PDH levels are high, what would you expect to be the consequence for circulating blood calcium?
Low.
Yeah, I just want you to understand, I want you to be thinking about this, because sometimes this can give us a hard time depending on how we appreciate it, which way we approach the problem. But the PDH cells are monitoring circulating calcium levels. And if calcium levels are low, what's going to happen to the concentration? What's going to happen? What are the PDH cells going to do in the parathyroid glands? If calcium levels are low, release PDH, all right? And then PDH is going to have what effect?
Increase the
Take from the
calcium.
bone and put the calcium into the blood. So I just want you to make sure you appreciate that fundamental relationship. Has a fairly short half-life, and so as the calcium levels increase, the PDH levels decrease. So it's getting kind of like that sawhorse or seesaw effect. There's a discussion in this slide about the regulation, how PDH regulates the osteoblast and osteoclast. And you have several little pictures here showing that, but let me just sort of try to simplify this as best I can. There is, in the general immunological literature, you often see symbols like this. This, for example, RANK, receptor activated nuclear something or another. This is an immune hormone. It's immunological. It's a cytokine, basically. I take that back.
All right.
It's a cytokine receptor. This is the whole point I was trying to make. I'm having a hard time with that. I'm still recovering from yesterday. Here's the point I'm going to try to make, and just bear with me. The L stands for ligand. All right. So this is the rank L is the hormone, or the immunological hormone type substance, in effect a cytokine. All right. So the point that I want you to get from this symbology is fairly common. What you will see, and we've seen all this, we've seen some of this already. For example, do you remember what R by R's are? Excellent, ryanodine receptors. So sometimes you'll see an L at the end of the abbreviation or the acronym, and sometimes you'll see an R. If you want to put an R here, you can, but I just want you to understand this is the hormone, or the chemical, the molecule that acts like the hormone, and this is the receptor, even though there's no R by it. All right. So if we have that clear, just be aware that this is a common symbology. In order for osteoclasts to do their job, in order for osteoclasts to be generated, remember they're formed from multiple precursor cells, rank L and rank have to get together. The receptor has to bind, the ligand has to bind to the receptor. Let me say that again. These two must form a hormone receptor complex in order for two things to happen. Osteoclasts to be formed from precursor cells, number one, and number two, for the osteoclast to go about the business of resorbing bone. All right. Normally, this is prevented by a molecule or a substance called OPG, osteoprotegerin. All right, so normally OPG is blocking the ligand from binding to the receptor. So OPG, we'll say for a moment, it acts as a competitive inhibitor. Not quite what it does. Yeah. Yeah, it binds to the receptor. Binds to the receptor. So it might be helpful to avoid the mistake that I made, is maybe imagine a little R there. I'm sorry, it binds the ligand. I'm sorry. See, I'm having, excuse me. It binds the ligand. But here's the important point. It binds the ligand. Here's the important point. When PTH is released, it binds, and this is what's very confusing, and this I do have right, despite your attempts to state otherwise, that PTH will bind to receptors on the osteoblast, not the osteoclast, but the osteoblast. So PTH binds to receptors on the osteoblast, and when that happens, the concentration of OPG, regardless of which one it binds, the concentration of OPG drops, and the concentration of RNKL, the ligand, increases. So let me say that again, because this is the only confusing part of this, and the reason you need to know it is because so many medications, so many osteoporosis medications are dependent upon understanding this principle. So the receptor for PTH, paradoxically, is not on the osteoclast, it is on the osteoblast. Okay, and when the PTH binds the osteoblast, several things happen. Most important of which is OPG levels go down, RNKL levels go up, and the net effect, as we indicated a moment ago, is to promote generation of osteoclasts and enhance their activity. So we have this rather bizarre indirect pathway by which the osteoblast is actually enhancing osteoclast activity. And the net effect then is bone resorption. Secondarily, PDH also enhances reabsorption. And notice the spelling here, reabsorption, not resorption, but reabsorption of calcium from the kidneys. And what would the effect of that be on circulating calcium concentrations? Increase. So this is another mechanism for increasing ECF calcium concentrations, and that's really what the objective here is, to increase the proportion of calcium that is in the blood as opposed to the bone. And PDH, as we've also pointed out in its chronicity with vitamin D, promotes uptake of intestinal calcium. So that too is going to fortify or increase the concentration of calcium in the blood. Vitamin D is a little bit simpler with respect to its, well, I shouldn't say that either. With respect to its ability to regulate blood calcium, it's a little simpler. It has, if you believe the literature, a broad spectrum effects on just about every aspect of your physiology, such that, as far as I can tell, if you get enough vitamin D every day, Did you hear that the oldest person died yesterday? We have a new oldest person, and they're a month younger than the previous oldest person. Yeah, 116, as I recall. Yeah, so as far as I can tell, if you have enough vitamin D, you'll live to be 120, 130. If you believe all the hype, if you believe all the advertisements. Part of the problem in understanding vitamin D is the terminology. And I'm not sure what to tell you here, except that we're going to find, and we'll just sort of work through this systematically, we're going to find that vitamin D is a steroid. It's a steroid-like chemical derived from cholesterol. And that cholesterol is going to be successively hydroxylated. There are a number of chemical modifications that occur in the process of generating active vitamin D from cholesterol. But the most important series of chemical reactions or chemical changes are these series of hydroxylations that occur in three different tissues. And they occur in this sequence: the skin, the liver, and the kidney. Now, I'm simplifying this a bit, but in general, we say that active vitamin D, or vitamin D3, is called calcitriol. Calcitriol, and suggesting three oxidation, or three hydroxylations. Calcidiol would suggest what? Vitamin D2, a precursor form, and so on. So when you look at your food, when you go home tonight and decide you want to be like that 116-year-old person living in, I don't know where they're living, I've not heard that.
Japan?
I thought it was Europe somewhere, maybe so. No, it's Japan. Tibet, Japan, same. So if you want to decide you want to live that long, what I would recommend is that you pick up some pastries at Publix or Winn-Dixie or whatever. Have you ever looked at the ingredients on the pastry packet?
Mm-hmm.
It just goes on and on and on and on. It's things in there that you wouldn't believe. I don't even think they originate on this planet. But you'll always see one of these two things, cholecalciferol or ergocalciferol, and these are all precursor forms. We talked about iodide being used to fortify, your foods being fortified with iodine. Milk is regularly fortified with vitamin D. Bread products, pastries, routinely fortified with vitamin D. I think even your Twinkies are fortified with vitamin D. But these are the precursor forms, and I just want you to be aware that these are relatively inactive until they've been acted upon by the liver and the kidney. So the terminology is extremely confusing. You'll see reference to calciferol, cholecalciferol, ergocalciferol, vitamin D2, vitamin D3, but all of these, with the exception of active calcitriol, are precursor forms. And remember, in natural, normal synthesis of vitamin D, three tissues are involved: the skin, the liver, and the kidneys. So the general effects of vitamin D physiologically, again, we've already pointed out most of these: enhancing intestinal uptake of calcium, to some extent phosphate, although the relationship can be a little tricky at times. And to a lesser extent, we've indicated that vitamin D can potentiate release of PTH. They sort of work together. And there's probably, to that extent, some influence of vitamin D on the kidneys as well. So notice the term here, and I just begin to sort of have you play with this in your head. It's not something I would ask you on a test. We call vitamin, one of these precursor forms, say D2 or ergocalciferol, a pre-hormone. Why would we use that term and not, for example, prohormone? What is a prohormone? Remember our discussion of the synthesis of peptide hormones? We begin with the pre-prohormone. Yeah. The pre-prohormone would certainly have the signal peptide. And all right, then we get down to if we chop off the signal, we would call that a what? Prohormone. So why don't we call this a prohormone? Well, it's certainly the molecule is the thrust here. Prohormones are by definition peptides. Is this a peptide? No, this is derived from cholesterol. So I just want you to be aware that there is a reason why this terminology is used. This is actually structurally a very different substance. So you do not need to understand the derivation from cholesterol to the vitamin D3, which I'm not even sure they show here. I guess this is the one they're showing here, but it does involve successive hydroxylations, again involving the skin, the liver, and the kidneys.
Can you supplement with the active form?
I don't think so. I don't think that's a good question. I've never really seen calcium. My guess is that if you did, it would have to be injected, and that might present issues of metabolism. And the concluding slide here is just to remind you, we talked about this as we talked about growth hormone. Growth hormone is certainly important for growth, but we also pointed out that thyroid hormone, cortisol, the sex hormones are also essential for normal growth. Likewise, I want you to appreciate the calcium regulation involves multiple hormones, and you shouldn't, in fact, we're going to talk about the way that multiple hormones regulate the blood sugar in just a moment. You can see virtually all the major families of hormones in one degree or another are involved in calcium regulation. This in particular is one you should pay attention to. We're going to talk about cortisol in just a moment. But one of the phrases you're going to learn in your pathophys is glucocorticoid-induced osteoporosis. Glucocorticoid-induced osteoporosis. So the implication there is that as cortisol levels go up, you would expect bone density to go down, all else being equal. And we'll talk more, when we get into reproductive, we'll talk about the contribution of sex hormones to bone development. As I say, since certainly well before the economic recession, we have known that vitamin D cures everything, every conceivable disease mankind is aware of. If you just have adequate levels of vitamin D, you need fear nothing. You can wrestle alligators and become president all in a single evening. So there's been a lot of hype. There was a panel, I thought it was 2012 or 2013. What is it? It was 2010, actually, before that. It was actually an FDA finding after this that said, well, maybe there is too much hype with respect to vitamin D. How many of you taking vitamin D supplements? Yeah. So I would have said probably 2 years ago that all the evidence that we could find would justify that. I talked earlier about the Copenhagen effect and the problems associated with taking multivitamin supplements, the increased risk of cancer and reduced longevity. There was a new study last week that pointed out how many people taking B vitamin supplements. Yeah. So now B vitamins are the rage, primarily because they help you withstand stress, something we're going to talk about in just a moment, and also ward off the risk of Alzheimer's. I don't know what to tell you. The science comes and goes, ebbs and flows. But certainly the FDA, this particular study was before that FDA report. I don't know if it's in here or not. Yeah, this is 2013. This was the FDA. Do not improve bone health. Low blood levels of vitamin D linked to chubbier kids, faster weight gain. It doesn't seem like that. This is troubling, isn't it? Fruit and vegetables do not reduce overall cancer risk. So vegetarian diet and lower your blood pressure. But in any case, with respect to vitamin D, you need to be aware of certain things like hypervitaminosis, excess vitamin concentrations, especially with fat-soluble vitamins A, D, E, and K. They become stored in the body in high concentrations and sometimes have adverse effects. You have to think a lot of it. In these studies with the B vitamins showing their beneficial effects, especially in lowering the risk of dementia, you'd have to take just enormous amounts of B vitamin to replicate those the findings that they had with animals. So I'm not sure what to tell you. It's a very complicated problem, but of course people are going to come to you and ask you, should they take vitamins? Should they take supplemental vitamin D? I don't know. You just have to peruse the literature.
But isn't most of the vitamins in order to absorb it, they need to be in fat. So if we drink water by taking taking multivitamins, actually most of it is going to be excreted
Well,
via
A, D, E, and K are your fat-soluble
right
vitamins. All
fat
the others are
soluble.
water-soluble. And you're right, the primary effect of vitamin supplements is to change the color of your urine.
Yeah.
But there are all sorts of salts and formulations now to enforce some of these vitamins. When, for example, you're going to be dealing with patients with anemia, you're going to inject them with the vitamins, and you're going to use other mechanisms to ensure high concentrations of the diet. Yeah.
I watched a documentary called Forks Over Knives, and they did. So you're familiar with that?
No.
Okay. Well, they did something called the China study, and through that they could look at purely what somebody was eating for protein-based diet, whether it was fish, chicken, red meat, whatever, and then they could pick out what kinds of cancer that they would have strictly from their diet. And then they said they quoted studies say that they could grow or basically kill cancer in rats and labs strictly by taking either 80% of their diet from a meat source protein or switching that 80% to a plant-based protein, and they could either grow or kill cancer based on that ratio and the acidity of the body.
Yeah, and of course you have to be a little skeptical of some studies. There's no doubt that protein-based diets are associated with higher cancer risk, especially in the GI tract and alimentary system, colon cancer, and so on. But sometimes that's you as a PA, you as a healthcare advisor, that may not be your principal consideration. Let's say you have an obese patient, the science is changing, and probably we should do something when we get to the GI tract. We'll talk about a recent finding that I think is very important for you folks. But in any case, you have an obese patient. What's the primary consideration there? Their long-term cancer risk, or maybe getting their weight load off? And so, it's been known for a long time that high-protein Atkins-type diets can be helpful in lowering the risk of diabetes. So I agree with you, but your job is a complex one, and you've got to weigh a lot of different factors to come to appropriate therapeutic conclusion.
Just for the vitamin D, I just did a project on this for the public health class, and currently three-fourths of the population is deficient in vitamin D, which just means like you have like less than 20 nanograms per milliliter in your blood, and they've done studies like even people in South Florida, there's I think only 10% of African Americans had adequate levels, and it was maybe 30% of Caucasian people, and it's been shown that if your blood levels are between, like you said, it might not matter for supplements, but if your blood levels are between 40 nanograms and 60 nanograms per milliliter, your risk of all-cause mortality goes down by an outrageous amount. So your chance of dying from anything pretty much goes down, and it's been shown to be involved in regulating, it regulates a 16th of your genome, which is an outrageous amount for one thing to do. So.
Well, there's certainly some degree of that, but again, should I believe you or the FDA?
I would believe me because I follow a doctor, and she spent her entire life studying this, and
there are no doctors at the FDA.
The current FDA recommendation.
Well, let's talk about that a second. What you had mentioned nanograms per unit of blood, which is a MASH unit, which is unusual. Normally, when you go to when you look at these fat-soluble vitamins, you get the supplements in the bottles. What is the normal unit there?
That's IU.
Yeah, it's international units. And a lot of that arose from the early part of the 20th century. There wasn't a lot of science based or formulation of those recommendations. And then two, because I've looked at a lot of the vitamin D literature over the years, a lot of the therapeutic doses that they recommend are just abnormal, just unrealistic. They would not be available or achievable in a normal diet, so they would require some degree of supplementation. You add that to the fact that when you go out in the sun, your skin is cranking out precursor forms of these vitamin D in terms of international units on the orders of thousands of units, and then it's rapidly resorbed. So the question from a biological perspective is, why is the skin doing that? Why is it generated? Why is it resorbed? Don't know. I don't know if we have this science to answer all those questions. But I would be a little bit skeptical of this vitamin D hype, simply because I could tell you, in 1999, what was the vitamin of the moment then? Every cardiologist in the country would have, well, that's always it. No, that was the Linus Pauling thing. In the late 1990s, every cardiologist in the country would have said that you should be taking vitamin A, because A had been shown over and over again to lower the risk of cardiovascular disease. And what was found a few years later? No, it's not true. In fact, it actually, vitaminosis actually increases the risk of cardiovascular disease. I worked in a free radical lab back in the 90s. What do we know about oxygen free radicals? They're bad, right? Just evil. And they're just ripping through your body right now, tearing you to pieces, just shredding every organ in your body. And what did we found? What did the science show? After all that hype, you could go into the library and find book after book after book. Have you found any disease? Do you know of any disease that has been directly implicated in a cause and effect fashion with the free radical levels, oxygen free radicals? Well, believe me, Alzheimer's, we talk about plaques, tau protein, synaptic. In all of these diseases, you always find reference to free radicals just because that's a nice thing to throw in. I mean, you always find implication. It's a theory of aging, for example. Anybody who's watched the X-Files. It's coming back here. So I would just encourage skepticism. I could tell you story after story, but yeah, I don't know that the science is absolutely clear on vitamin D, but I'm not sure that I believe all the hype in light of recent literature. All right, we have a few minutes. I want to then begin at least a discussion of the adrenal glands. The adrenal gland is going to be very important for you because the adrenal gland, sitting as it does atop the kidneys they're sometimes called the suprarenal, especially in the European literature. The adrenal gland consists of two fundamental tissue layers. There's an outer cortex, and this is going to be the topic of most of our discussion, from which the adrenocortical hormones are released. And I think we made the point earlier that these adrenocortical hormones are chiefly steroids that follow pretty much the same synthetic pathway as that occurs in the gonads, the testes and the ovaries. And then interior to that is the adrenal medulla, which is, we've talked about this when we talked about the sympathetic nervous system. This is basically the sort of the post-ganglionic neural tissue for the sympathetic nervous system. When activated, when stimulated, it releases adrenaline into the general circulation. Now, one of the things you need to be able to do for the next test is to, and I would encourage you to maybe find some better illustrations. I didn't necessarily give you the best illustrations here, but primarily because I'm more interested in you knowing the sequence of tissue layers here. So we're looking at the adrenal cortex. These pictures are upside down, by the way, because I want to make sure you just don't memorize for the test top and bottom and go through the list. Over on the right here, this is the outside. Over on the left, this is the outside. So this is the exterior of the adrenal gland. On the left, this is the exterior in this illustration. And so over here, for example, here's the adrenal medulla, and this would be the capsule, adrenal capsule. And as I say, we're primarily focusing upon the adrenal cortex right now, and you'll notice that there are three general histological layers that are over here. You need to know the names of those layers: zona glomerulosa, fasciculata, and reticularis. And sometimes you see the histological basis for this distinction, sometimes you don't. I'm going to return to that theme in just a moment, but by way of introduction, these three zones are associated with those three fundamental categories or classes of adrenocortical steroid. If you come down here in this picture, again, outsides at the bottom, the zona glomerulosa is primarily associated with release of mineralocorticoids such as aldosterone, these hormones that regulate renal function, for example. The middle, I hesitate to say interior, but the middle region called the fasciculata is the primary region responsible for release of the glucocorticoids such as cortisol. And then adjacent to the medulla is an adrenocortical zone called the zona reticularis that is responsible, and this is going to be very important in our discussion of reproductive physiology, that releases sex hormones. So all three are right there together. Why don't we stop there, take a break, and then we'll talk about more about this zonation.